All right, so we spent a good chunk of time talking about how things are being filtered here in, or by the glomerulus and caught by the glomerular capsule. Let's spend a little bit more time talking about what's actually happening now in these tubes. So in this image, you're seeing the, again, glomerular capsule, the proximal convoluted tubule, the descending limb of the loop of Henle, the ascending limb of the loop of Henle, the distal convoluted tubule, and then the collecting duct, and where the collecting ducts kind of come down to this one final point, that's called the papillary duct. Now, one nice thing about this image is it really stretches a nephron out, so it's nice and linear. As you saw in earlier diagrams, like there is some overlap between the distal convoluted tubule and even this afferent arteriole. So this is more of an idealized image trying to help show you the different components and the flow of this tubule portion. Okay, so just a couple of terms for you. There's reabsorption, so not absorption, because the only time we have absorption is our digestive system. So the first time we absorb it from the lumen of the digestive system into our bloodstream, that is being absorbed. Anything that we then filter and then have to take back is now reabsorption. So it's going back into the bloodstream. There's two ways that things can be reabsorbed. They can either be done paracellularly, meaning between cells, or transcellularly, meaning they're moving across the cell. So it's going through the actual cell itself, not just between two of them, which is paracellular, but actually through the cell, right, into the bloodstream. And then secretion is just the opposite. As I mentioned to you before, not everything gets filtered that we initially want to have filtered. So then we actually have to actively move material into a tubule. Because we're actively moving it and it's not just being kind of filtered like in a colander like we saw with the strawberries, it is now called secretion. So again, it's not actively being filtered like a colander. So we're actually, there's intention and active transport of materials. It's considered secretion. All right, so first, the proximal convoluted tubule, and we'll go in order of these tubules. The vast majority of reabsorption is going to occur in the proximal convoluted tubule. So the most of the water, sodium, potassium is being reabsorbed here, although not all. 65%, while a clear majority, that's still another 35% that needs to be absorbed. It's still quite a bit of water that needs to be reabsorbed later on in the tubule network. Small ions can be reabsorbed, things like chloride ions, magnesium, calcium, potassium. All of the glucose will be reabsorbed. Glucose is a small molecule, so it's easily filtered. But we do have symporters, so these are active transporters that are going to move glucose back into the bloodstream. However, they have an upper limit of how fast they can work. Another way of saying that is that each one of these symporters has a transport maximum. So kind of like that image of I Love Lucy that I talked to you about with the chocolate factory, each one of these symporters can only work so fast. So meaning, like in diabetics who may have glucose present in their urine, that means that there's so much glucose in their bloodstream that those little symporters have hit their transport maximum and they're taking up all the glucose they can, but there's still so much more that it's still being lost in the urine. Now, under non-diabetic conditions, under normal healthy conditions, our symporters can easily pick up the glucose that's in our bloodstream. Only under very rare conditions, like if you ate like a bag full of candy corn in one fast sitting or something where you have a massive spike in sugar levels, might they be overwhelmed. But under normal conditions, healthy conditions, these symporters will reabsorb all of our glucose. And some vitamins may also be reabsorbed. Now, in our loop of Henle, in the descending limb, it's only permeable to water. So the only thing that is going to be reabsorbed in the descending limb of the loop of Henle is going to be water. Now, on the other side of it, in the ascending limb, it's almost exactly the opposite. No water gets through, which is amazing. because I think we tend to think of like water can go through anywhere. No, it cannot. You have to have aquaporin channels. It just so happens that almost every cell in the body has aquaporin channels that allow water to get through it. However, if they are not inserted into the cell membrane, water cannot get across. So one of the few places in the body where they are not found is going to be in this ascending limb of the loop of Henle. So the only thing that's being reabsorbed here is chloride, potassium, and sodium. As we move into the early distal convoluted tubule, we'll also have the transport of sodium back into the cell, which means that we then lose potassium. We have continued water reabsorption. So again, if we had about 65% in that proximal convoluted tubule, we had a little bit more in the loop of Henle. We have another about 10 to 15% here in the early distal convoluted tubule. So we're up to somewhere around 80-85% of the water is reabsorbed here, or by now has been reabsorbed, I should say, not here. Again, here, just 10 to 15, but by the time we get here, most of the water has been reabsorbed. Calcium is also reabsorbed. We do need to make sure, but this is going to be dependent on parathyroid hormone levels. So if there's not enough parathyroid hormone levels being secreted, calcium will not be reabsorbed. And then some medications and drugs will be secreted into the distal convoluted tubule. Penicillin is one such medication. And then in the late distal convoluted tubule and the collecting duct, these are more similar in their function. So there's a little bit of a difference in the function of the early distal convoluted tubule versus the late distal convoluted tubule, the late one being closer to where it's going to connect to the collecting duct. These two parts of the tubule system have similar cells called principal cells and then intercalated cells. Principal cells are going to reabsorb sodium and secrete potassium, and that's done via the sodium-potassium pump. So as the principal cells are reabsorbing sodium, the intercalated cells then go, all right, well, heck, we need that potassium. We don't need to lose that in the urine. So they will then reabsorb potassium and bicarbonate, but they will secrete hydrogen. Okay. By the time we get to this distal convoluted tubule now, and by the way, there's aquaporin channels here too. We have reabsorbed vast majority of the solutes and the water, and the principal cells in the collecting duct will make the final adjustments. So, if I can summarize what's going on in these tubules, the main things to kind of keep in mind is that the vast majority of reabsorption is going to happen here in the proximal convoluted tubule. Most of the water, 65%, is being reabsorbed here in the proximal convoluted tubule. Only water is being reabsorbed here in the descending limb. Only potassium, chloride, and sodium here in the ascending limb. In the distal convoluted tubule, we'll have some more water reabsorption, sodium, chloride, you can see this here, calcium, and then here in the late distal convoluted tubule, as well as the collecting duct, we have our water, our sodium, our potassium, and then we'll also have bicarbonate being reabsorbed and secretion of hydrogen ions. As far as these percentages go, you don't have to memorize all of these. That's too much information. Again, vast majority of water here. By the time we get to late, almost all the water has been reabsorbed, the last bit being concentrated here in the collecting duct and the distal convoluted tubule. Okay, hopefully that makes sense, and then we're going to move on to how we do some of this concentration.